Uncoupling proteins are a subfamily of the mitochondrial carrier family. They are located on the inner mitochondrial membrane, where they dissipate the proton gradient generated by the activity of the respiratory chain, so uncoupling mitochondrial respiration from oxidative phosphorylation. This process is well known in non-shivering thermogenesis in brown adipose tissue. Recent evidence has suggested that some members of the uncoupling protein family are able to transport metabolites across the inner mitochondrial membrane, so their functional role as solute transporters has to be further clarified. In this regard, we have studied UCP4A, a member of this family, in Drosophila melanogaster, by highlighting that this protein has a central role in signal transmission in the central nervous system. Previous studies highlighted that this protein has a protective effect on mitochondrial function. In particular, its expression can rescue locomotor defects, muscle degeneration, and can also prolong survival of dopaminergic neurons in Parkinson's disease models in the fruit fly. But the biochemical mechanism through which all this is possible was not known. So, in order to investigate these aspects, we decided to identify the sequence of this protein by sequence homology, and then we proceed to the amplification, cloning, and overexpression in heterologous systems of our protein in order to study its transport activity. Here you can observe the overexpression of UCP4A in bacterial cells. In these cells, this protein accumulates as inclusion bodies. We have partially purified it by sucrose gradient centrifugation. Then we have solubilized it and used it in functional reconstitution into liposomes in order to test its transport activity. Here you can observe a scheme of this process. First of all, we prepare a reconstitution mixture containing the solubilized recombinant protein. Then we perform several chromatographic steps in order to obtain proteoliposomes loaded with a cold substrate and containing the recombinant protein in their membrane. In this condition, we can start an exchange reaction by adding from the outside a radiolabeled substrate. The exchange reaction can be stopped by the external addition of two irreversible inhibitors, pyridoxal phosphate and beta-phenanthroline. Then we can remove the external substrate by a further chromatographic step, and then we can count the radioactivity inside proteoliposomes, which gives us a measure of the transport efficiency of our protein. In the same way, we can study uniport reaction, but in this case we don't use substrate inside, only sodium chloride, and also in this case we need to isolate and count protoliposomes and count radioactivity inside. According to these two procedures, we have studied in forward mode the exchange and the uniport reaction catalyzed by our recombinant protein. So this protein is able to exchange aspartate, and also it can catalyze a uniport reaction. in order to fully investigate these two different reactions, we have used another approach. We have used the backward mode. This means we have prepared proteoliposomes with internal radiolabeled substrate. This is possible by carrier-mediated exchange equilibration. This means that at the end of the process you have just seen, we don't add PLP and beta-phenanthroline, but only a reversible inhibitor, that is the PCNBS. In these conditions, we are ready to start the exchange reaction by adding from the external side cold aspartate together with DTE, which can reverse the inhibitory effect of PCNBS. And so, in these conditions, we can observe the efflux of radiolabeled aspartate from the inside towards the external side of our proteoliposomes. When we want to study the uniport reaction, we have to add from the external side DTE and a buffer. In this case, we have used two different buffers. The first at a pH value of 6.5, which is the same pH value present inside the proteoliposome. Then we have used a buffer at a pH value of 8. And as you can see, when we have imposed a delta pH across the proteoliposomal membrane, we cannot observe a significant difference between these two curves. This means that our transport is not proton dependent, but only aspartate can move, so our transport is electrogenic. Then, by the use of further experiments, by varying the external radiolabeled concentration of aspartate, we have measured the kinetic constants of our recombinant protein, as you can see in this slide. Then we have tested substrate specificity and inhibitor sensitivity of our protein. In the first case, we have used hetero exchanges. This means radiolabeled aspartate outside and one cold substrate inside protoliposomes. We have used each one at a time, in protoliposomes. And so you can see that the aspartate is the best transported substrate. And to a lower extent, our recombinant protein can also exchange these other substrates. For inhibitor sensitivity, we have performed HOMO exchanges. This means the same substrate inside and outside. We have used aspartate, but together with the external radiolabeled aspartate, we have added a cold substrate, each one of these molecules. And so also in this case, you can observe that cold aspartate can inhibit the entry of radiolabeled aspartate. On the other hand, you can observe the powerful inhibitory effect exerted by all of these well-known powerful inhibitors of many mitochondrial carriers. Then we have tested transport ability of our recombinant protein by mitochondrial swelling experiments. Mitochondrial swelling can occur when large amounts of a solute can enter mitochondrial matrix. This is possible only when we have a charge-balanced transport. And in order to achieve this aim, we have two options. The first one occurs when we have a proton-assisted transport. In this case, we could observe a mitochondrial swelling when we use an ammonium salt of the anion we want to test. But this is not our case, because we have just seen that our transport is not proton-assisted. The other option is when we have a not proton-assisted transport. In this case, we have to use the potassium salt of the anion we want to test, together with valinomycin, that is able to transport into the mitochondrial matrix potassium ions. So, by using these methods, we have isolated yeast mitochondria of 3 different yeast strains. In the first case, we have the wild-type strain, and you can observe 2 different swelling. The first one is due to the presence of the phosphate carrier, which is able to co-transport into the mitochondrial matrix phosphate together with a proton. The second one is due to the presence of the aspartate glutamate carrier, which can co-transport into the mitochondrial matrix aspartate together with a proton. When we make a deletion of the aspartate glutamate carrier, we create a deletion strain, and its mitochondria are no more able to catalyze the entry of ammonium aspartate, as you can see here, because aspartate glutamate carrier is absent in this condition. When we express in the deletion strain our recombinant protein, we can note a mitochondrial swelling due to the presence of our protein. Note that this swelling occurs when we add valinomycin. And so we can say that our recombinant protein is able to unidirectionally transport only aspartate. In this regard, you can note that the other potassium salts are not transported into the mitochondrial matrix. Our transport is not proton-coupled. In this regard, you can note that ammonium aspartate is not able to cause mitochondrial swelling, and aspartate very probably is transported outside the mitochondrial matrix. This is because we have a negative potential inside the mitochondrial matrix. So this direction could be promoted by the electrical gradient present across the inner mitochondrial membrane. But in order to test this hypothesis, we have used yeast cells. You have to know that yeast cells are able to grow on oolate only when they have a functional malate aspartate shuttle. This is required for the reduction of cytosolic NADH derived from the oxidation of oolate. But when we have the deletion of the aspartate glutamate carrier, we interfere with the function of the malate aspartate shuttle, and so the deletion strain is no more able to grow on oolate, used as a sole carbon source. In yeast we have also another transporter, YMC2. This is able to import glutamate plus a proton into the mitochondrial matrix. So our hypothesis is that if our recombinant protein can really export aspartate from the mitochondrial matrix towards cytosol, and this transporter can work together with YMC2. Their combined activity can re-establish the transport catalyzed by the aspartate glutamate carrier, which is able to import into the mitochondrial matrix glutamate plus a proton and is able to promote the exit of aspartate from the mitochondrial matrix. And so, if we have this situation, we can rescue the growth defect on OLAT in the deletion strain because we will have a functional mass. We have successfully tested this hypothesis by using 4 different yeast strains. For the wild-type strain, you can observe that this strain has no problem in growing on liquid medium containing OLAT as a sole carbon source. On the contrary, for the double mutant strain, but I had also for the strain lacking the aspartate glutamate carrier gene, we have no growth on OLAT, as you can observe here. Even when we transform the deletion strain with the gene of our recombinant protein, we do not observe the growth because this is not enough to re-establish the activity of mass. But when we co-express both proteins in the double mutant strain, in this case we have re-established the activity of the aspartate glutamate carrier and also that of the malate aspartate shuttle, and so we can observe a growth. This means that our protein is able to export aspartate from the mitochondrial matrix towards the cytosol, and so this is possible also in vivo conditions. Next, the aspartate transported to the cytosol can be used in the biosynthesis of proteins and nucleotides, and also in the biosynthesis of beta-alanine and N-acetyl aspartate. These two molecules are very important in neurotransmission. The first one plays a key role in the recycling of biogenic amines, histamine and dopamine. In other words, these molecules take part in reaction aimed to the transport of these neurotransmitters to presynaptic neurons. And so, this is a very important consideration in neurotransmission. From the other side, we have an increased biosynthesis of N-acetyl aspartate that is a precursor of myelin biosynthesis. And of note, in Parkinson's disease, also in humans, a decrease in the level of these compounds was found. And so on this basis, we can understand why when we express the UCP4A protein in fruit fly models of Parkinson's disease, we can improve the phenotype of the fruit fly by rescuing locomotor defects, as shown in a previous slide. So this is a very important consideration. In the future, we intend to continue the study on aspartate and to further investigate on the specific metabolic pathways in which this molecule can be involved in order to shed light on the molecular basis of neuromuscular diseases. If you are interested in this topic, you can contribute through this special issue for which I serve as a guest editor. Thank you very much for your attention.